Myocardial autorhythmic cells are the impetus for the excitability of myocardial contractile cells. So what we've talked about in previous videos is that the heart muscle, the myocardium, has two main types of cells: contractile cells, which are going to allow for the pumping action of the heart, the contraction of ventricles to pump blood either to the lungs or to the systemic circulation. But there are also myocardial autorhythmic cells, and these are cells that spontaneously depolarize and repolarize and depolarize and repolarize constantly without ever stopping. And they set the basis for heart rate. Full disclosure, the heart does have nervous stimuli and receptors for neurotransmitters and hormones that modulate or modify these autorhythmic cells. So if we ever need our heart rate to increase during a sympathetic response, the myocardial autorhythmic cells will increase the rate of firing. Or if we are exhibiting parasympathetic activity, otherwise known as vagal tone, it will decrease the rate of myocardial autorhythmic cells. In this video, we're just going to talk about how myocardial autorhythmic cells maintain their spontaneity. So in this slide, we're looking at the graph for myocardial autorhythmic cells, which I tend to abbreviate as MACs. And threshold is going to be about negative 40 millivolts. I'm not super concerned with what the actual voltage is, but most textbooks are going to suggest it's negative 40 millivolts. And we are going to have an initial slow depolarization of these MACs that is referred to as a pacemaker potential. So there is no resting membrane potential in MACs. That is replaced with a pacemaker potential. And as we can see, that is slowly exhibiting a depolarization event. That's initiated by funny current channels, abbreviated IF, and they were initially described as funny current channels. I represents current, but funny because the initial researchers really didn't understand what was going on. And these are similar to nicotinic channels at the motor end plate of skeletal muscle fibers that are monovalent cation channels. That is to say, they allow the passage of sodium and potassium. And as we've learned previously, when both those cations pass through the same channel, sodium has a bigger impact on the resting cell because it's moving down its electrochemical gradient, and potassium is only moving out of the cell via its chemical gradient. And then the rest of this pacemaker potential is finished off by some calcium channels. Influx of calcium continues this depolarization event. So let's just suggest the first half is the result of these funny current channels with sodium coming in, to initiate the depolarization. And then that is finished off by a little bit of calcium coming in to continue that depolarization up to threshold or up to negative 40 millivolts. That is going to open up a number of other calcium channels to cause this big spike to initiate the full-on action potential of the MACs. So now we have a massive influx of calcium moving into the MAC, which is going to depolarize the cell, let's say up to roughly 10 millivolts. And then repolarization is going to be typical as in other cells where it's going to be potassium efflux. So here, once again, we're back down to negative 60. And because I've suggested that the myocardial autorhythmic cells do not exhibit a resting state, we're going to drop right back into this pacemaker potential due to the opening up of the funny current channels and then the opening up of a few calcium channels, then a huge spike in this depolarization due to large increase in permeability to calcium. We're going to get repolarization due to potassium efflux, and that's going to keep going and going and going. And it's theoretically never going to stop. Certainly there are individuals who have problems with their electrical activity or their electrical conduction system of the heart. The myocardial autorhythmic cells are going to have an action potential roughly 80 to 100 times per minute. For our purposes, let's just say it's 80 times per minute. Now, generally speaking, most people have a resting heart rate lower than 80 times per minute. Let's say it's 60 times per minute. Certainly, this is highly variable. The resting heart rate is the result of parasympathetic activity influencing the myocardial autorhythmic cells, specifically causing the myocardial autorhythmic cells to slow down. So the baseline for myocardial autorhythmic cells is roughly 80 to 100 times per minute. Some books are going to suggest 90 times per minute. Just for our purposes, let's just throw out the number 80. 80 times per minute, we are going to get peaks 80 times per minute. A depolarization, repolarization, depolarization, repolarization. There is never a resting state for myocardial autorhythmic cells. So in closing, I will tell you that the heart does not need nervous stimuli or hormonal influence to cause the heart to beat, to initiate excitability of myocardial contractile cells. But I will also tell you that the MACs almost always have some sort of endocrine or nervous stimuli modifying their rate. And we'll talk about how that's achieved in the subsequent video.